All right, get ready, because today we're going deep into hedgehog signaling.
Hedgehog signaling.
It's pretty amazing.
It is.
This cellular communication system that's vital
it is.
for embryonic development.
Absolutely.
And how adult stem cells work.
It's really remarkable. Imagine a tiny little embryo, just a cluster of cells, and it somehow transforms into this complex organism with all these intricate parts.
Right.
Hedgehog signaling is like one of the key players.
So we're talking about cells talking to each other.
We are.
But what are they saying?
Yeah.
How does this hedgehog thing fit in?
Well, think of it like this.
Okay.
Cells need instructions.
Yeah.
They need to know what to do, how to grow, where to go.
Oh, okay.
Hedgehog signaling provides those instructions.
Okay. It's like...
So it's like a GPS.
Like a cellular GPS system.
Oh.
Guiding cells to their proper destinations.
Telling them what kind of cells to become.
So what does the hedgehog part even mean? Is it some kind of spiky signal? Zipping between cells?
Not quite spiky, but the name does have a kind of funny origin.
Okay.
It's named after a family of proteins, one of which is called Sonic Hedgehog.
Oh.
Or for short.
Sonic Hedgehog. Like the video game character.
Exactly. Scientists have a sense of humor, too, you know?
Yeah.
But don't let the name fool ya. Shh is a serious player in this process.
Okay.
It's a signaling molecule or ligand that acts as the messenger.
Oh.
Carrying instructions from one cell to another.
So shh is the message.
Yes.
But who's receiving it?
That would be the patched receptor, or Ptch, another protein, sitting on the surface of the receiving cell. Okay. Think of Ptch as the mailbox, waiting for shh to deliver its message.
Okay, so I'm picturing shh, speeding over to Ptch, message in hand. What happens next?
This is where it gets interesting.
Okay.
Sh binding to Ptch actually inhibits Ptch's normal function.
What?
Which is to suppress another protein.
Oh, wow.
Called Smoothened.
Smoothened?
Or Smo.
Smoothened. Okay. So it's like sh is taking the brakes off Smo.
That's a great way to put it.
By binding to Ptch.
With Ptch out of the way, Smo is free to become active and set off this chain reaction that ultimately leads to the activation of GLI transcription factors.
GLI transcription factors. Those sound important.
They are.
What do they do?
GLI proteins are like the foreman of a construction crew.
Gotcha.
They receive the message from SMO and then head to the cell's nucleus where they control which genes are turned on or off.
Wow.
These genes are the blueprints that dictate what the cell will become and how it will behave.
So sends a message, Ptch receives it, SMO gets activated, and then the GLI proteins go and turn on. The right genes. It's like a cellular relay race.
That's a good analogy. And this relay race is happening constantly during embryonic development, ensuring that cells differentiate into the right types and form the right tissues at the right time.
This is fascinating. But I have to admit, it's getting a bit complex.
Sure.
Can we break down this pathway step by step, starting from the beginning?
Let's start with shh. Our speedy messenger, is produced by certain cells and diffuses outward, creating a concentration gradient.
A concentration gradient? What's that?
Think of it like ripples spreading out from a pebble dropped in a pond.
Okay.
The concentration of shh is highest near the source and gradually decreases as you move further away.
So it's not just about whether shush is present or absent. It's about how much of it there is in a particular location.
These gradients are crucial because different concentrations of shh trigger different responses in cells, leading to the formation of distinct tissues and organs.
Okay, I'm starting to see how this could work. But how do cells actually read these shh gradients? Is it like they have little measuring sticks?
Well, they don't have measuring sticks.
Okay.
But they do have Ptch receptors, remember? The number of Ptch receptors on a cell's surface influences how sensitive it is to shh.
So a cell with lots of Ptch receptors will be more sensitive to lower concentrations.
Exactly. And that's how different cells can respond differently to the same shh gradient, depending on their location and the number of Ptch receptors they have.
All right, so we've got shh traveling through the embryo, creating a concentration gradient. Cells are sensing this gradient with their Ptch receptors. What happens next?
Well, once shh binds to Ptch, it triggers a series of events that ultimately leads to the activation of SMO, as we talked about earlier.
Yeah.
But the way SMO gets activated is actually quite fascinating.
Okay, I'm all ears. Tell me more about this smoothened protein and how it gets turned on.
So in the absence of
Okay.
shish keeps SMO sequestered away from a special cellular structure called the primary cilium.
The primary cilium. That sounds familiar, but remind me what it is.
It's a tiny hair-like projection.
Oh, right.
That extends from the surface of many cells.
Okay.
It acts like a cellular antenna, sensing signals from the environment.
So Trish is keeping SMO away from this antenna, preventing it from picking up any signals.
But when binds to Ptch, it releases SMO, allowing it to move to the primary cilium.
Okay.
And once SMO is at the cilium, it can interact with other proteins and set off the downstream signaling cascade.
So the primary cilium is like a signal.
That's a great way to think about it.
Processing center for hedgehog signaling.
It's where the message from gets amplified and translated into action.
This is all incredibly intricate.
It really is remarkable.
It's amazing to think about. All these tiny proteins interacting with each other, orchestrating such a complex process.
And we've only just scratched the surface. There are many other layers of complexity to this pathway we could delve into.
Let's do that. I'm ready to dive deeper into the world of hedgehog signaling.
All right, so we've got Smoothened at the primary cilium, ready to pass the baton in this cellular relay race.
Yeah.
But what happens next?
Yeah, that's a great question. It feels like we've zoomed in on this tiny part of the cell, this primary cilium. And now I'm curious about what's happening inside it.
Well, the primary cilium isn't just a passive antenna. It's a dynamic hub of activity. When SMO arrives at the cilium, it interacts with a whole cascade of other proteins, setting off a chain reaction. That ultimately leads to the activation of GLI proteins.
So it's like a Rube Goldberg machine.
That's a pretty good analogy.
Of molecular interactions. One protein activates another, which activates another, and so on.
And the end result of this intricate cascade is the release of GLI proteins from a complex that normally keeps them inactive.
Okay, so the GLI proteins are finally free to do their job. They head to the nucleus, the cell's control center, and start turning genes on and off.
Yes.
But what kind of genes are we talking about? What are they actually controlling?
The genes regulated by GLI proteins are involved in a wide range of processes. From cell growth and division to cell fate determination and tissue patterning, they're essentially the blueprints that guide how cells behave and what they become.
So depending on which GLI proteins are active and which genes they turn on or off, a cell might become a muscle cell, a nerve cell, or something else entirely.
Exactly. And that's how hedgehog signaling can orchestrate the development of such a diverse array of tissues and organs. From a single fertilized egg.
This is mind-blowing. It's like a single pathway is responsible for creating all the different parts of our bodies.
It really is a remarkable system.
It is.
And it's not just about embryonic development. Hedgehog signaling continues to play a crucial role in adults, particularly in tissues that undergo constant renewal.
You mean like our skin and hair?
Exactly. And our gut lining. And even our blood.
Wow.
These tissues rely on stem cells, which are like the body's master cells, capable of self-renewal and differentiation into various cell types. Hedgehog signaling helps regulate stem cell behavior, ensuring that tissues can regenerate and repair themselves.
So it's like hedgehog signaling keeps
it's a great way to put it.
The construction crew on call, ready to replace damaged cells. And maintain the integrity of our tissues.
It's this constant balancing act, ensuring that stem cells divide and differentiate at just the right rate to keep our tissues healthy and functioning properly.
But what happens when this balance gets disrupted? We've talked about how important,
unfortunately,
hedgehog signaling is.
But
can things go wrong?
As you might imagine. A pathway as crucial as this one needs to be tightly regulated. Too much or too little hedgehog signaling can have serious consequences.
Like what? What kinds of problems can arise when hedgehog signaling goes awry?
Well, during embryonic development, disruptions in hedgehog signaling can lead to a variety of birth defects. One example is holoprosencephaly. The severe brain malformation that occurs when the forebrain fails to divide properly.
That sounds really serious. And what about in adults? What happens when hedgehog signaling gets messed up later in life?
In adults, overactivation of the hedgehog pathway is implicated in various types of cancer.
Oh, wow.
Including basal cell carcinoma.
Basal cell carcinoma.
The most common form of skin cancer and some brain and blood cancers.
So in those cases, it's like the signaling pathway is stuck in the on position, causing cells to grow and divide uncontrollably.
It's like having a construction crew that keeps building without any blueprints or oversight.
Wow.
The result is a chaotic mass of cells, a tumor, that can disrupt the normal function of tissues and organs.
It's amazing how a single pathway can have such profound effects on both development and disease.
And it underscores the importance of understanding this pathway in its full complexity. By learning how hedgehog signaling works, we can gain insights into both normal development and the development of diseases. And that knowledge can ultimately lead to the development of new therapies.
Speaking of therapies, you mentioned earlier that hedgehog signaling is a potential target for drug development. Can you tell me more about that?
Researchers are exploring two main approaches to targeting hedgehog signaling. Agonists, which activate the pathway, and antagonists.
Antagonists,
which block it.
So agonists would essentially turn up the volume.
That's a good way to think about it.
Of hedgehog signaling, while antagonists would turn it down.
And the choice of whether to activate or block. The pathway depends on the specific therapeutic goal.
Can you give me some specific examples? When might you want to activate the pathway? And when might you want to block it?
For conditions where stimulating cell growth and regeneration is desired, such as in wound healing or spinal cord injuries, hedgehog agonists could potentially be beneficial. They might help promote tissue repair by activating stem cells and encouraging them to differentiate into the necessary cell types.
That makes sense. And what about antagonists? When would those be useful?
Hedgehog antagonists are being investigated as potential treatments for cancers driven by overactive hedgehog signaling by blocking the pathway. These drugs could potentially halt the uncontrolled cell growth. That characterizes cancer.
So it's like applying the brakes to that runaway construction crew we were talking about earlier.
The goal is to restore balance to the pathway. Either by boosting it when it's too low or dampening it when it's too high.
This is all incredibly exciting. It sounds like manipulating hedgehog signaling has the potential to address a wide range of medical needs. But I imagine there are also challenges involved.
You're absolutely right.
It's not as simple as just flipping a switch. On or off is it?
Targeting a pathway as fundamental as hedgehog signaling requires a nuanced understanding of its complexities and potential side effects.
What kind of side effects are we talking about? What are some of the challenges researchers are facing in developing these therapies?
Well, one challenge is tissue specificity. Hedgehog signaling plays different roles in different tissues. So a drug that activates the pathway in one tissue might have unintended consequences
in another.
So it's like you want to deliver the message to a specific address, but you risk it being intercepted by other cells.
Researchers are working on ways to develop drugs that specifically target hedgehog signaling in the tissues of interest, minimizing off-target effects.
What are some other challenges? Are there concerns about long-term effects?
Yes, that's another important consideration. Hedgehog signaling is involved in many processes throughout life. So long-term modulation of the pathway could have unforeseen consequences. Researchers are carefully studying the effects of these drugs to ensure their safety and efficacy.
It sounds like there's a lot of exciting research.
It really is a testament to the power of scientific discovery. And it's important to remember that this is still a relatively young field. So much more to learn about hedgehog signaling and its potential applications. This deep dive has given me a whole new appreciation for the complexity and elegance of cellular communication. It's incredible to think about all these tiny proteins working together to shape our bodies and keep us healthy. It really makes you realize how much is going on inside us at the microscopic level, things we never even think about. But that are so crucial for our health and well-being.
It's true.
We've talked about how hedgehog signaling can go wrong, leading to birth defects and cancer. But are there other areas where research is focusing on this pathway.
Definitely. There's growing interest in exploring the role of hedgehog signaling in tissue regeneration and repair.
So could we potentially harness this pathway?
Yeah.
To help heal wounds faster? Or even regenerate damaged organs?
That's the hope. Researchers are investigating whether hedgehog agonists could be used to stimulate stem cell activity and promote tissue regeneration in conditions like spinal cord injuries, heart attacks, and even neurodegenerative diseases.
It's amazing to think that we might one day be able to tap into this ancient signaling pathway to repair damage that was once thought to be irreversible.
It's a truly exciting area of research with enormous potential to transform medicine.
Wow.
However, there's still much to learn before these therapies become a reality.
What are some of the key questions that researchers are still trying to answer?
Well, one key question is how to precisely control hedgehog signaling, as we discussed earlier. The pathway needs to be carefully balanced. Too much or too little can have negative consequences. Researchers are working on developing ways to fine-tune hedgehog signaling, delivering the right amount of activation to the right cells at the right time.
That sounds like a delicate balancing act. Are there other challenges that need to be addressed?
Another challenge is understanding the long-term effects of modulating hedgehog signaling. As we learn more about the pathway's roles in various tissues and processes, we need to ensure that therapies targeting hedgehog signaling don't have unintended consequences down the road.
So safety and efficacy are paramount.
Absolutely.
As with any new medical treatment.
Rigorous research and clinical trials are essential to ensure that these therapies are both safe and effective for patients.
It's fascinating to think that something as seemingly simple as a signaling pathway between cells could hold the key to treating such a diverse range of conditions.
It really speaks to the interconnectedness of biology.
It does.
Often what appears simple on the surface is incredibly complex and nuanced when you delve deeper. And that's what makes scientific discovery so fascinating.
Well, this has been an incredible deep dive into the world of hedgehog signaling. We've covered a lot of ground.
We have.
From the basic mechanism of the pathway to its roles in development, disease, and potential therapeutic applications. We've seen how this single pathway can have profound effects on our body, shaping us from the earliest stages of life and influencing our health throughout adulthood.
What are some key takeaways that you hope our listener will remember from this deep dive? I'd say the main takeaway is that cellular communication is essential for life. These intricate signaling pathways, like the hedgehog pathway, orchestrate a symphony of cellular events that keep us functioning properly.
And when these pathways go awry, the consequences can be significant, leading to developmental disorders, cancer, and other diseases.
But by understanding these pathways, we can develop new strategies to diagnose, treat, and even prevent these conditions. The research happening in this field is incredibly exciting.
It is.
And holds immense promise for the future of medicine.
As we wrap up this deep dive, I'm left with a sense of awe at the complexity and beauty of the biological world. It's a reminder that there's always more to learn.
Absolutely.
More mysteries to uncover. And it underscores the importance
of supporting scientific research. The discoveries made in labs today have the potential to transform lives tomorrow. So to our listener, keep asking questions, stay curious, and never stop exploring the fascinating world of science. Who knows what incredible breakthroughs await us just around the corner?